Hello, folks. This is Dr. Tarun. I will be providing all the lecture videos for this unit. So this is the beginning of Unit 3. In this unit, we will continue to explore the complex structures and diverse functions of the cell, extending our focus into various membrane-bound compartments inside the cell. Here in section 5.3, our main learning objective is for you to be able to describe at the end of this video portion the key features that distinguish prokaryotes from eukaryotes. The sub-objectives are twofold. The first is to be able at the end of this video to describe whether prokaryotes have internal membrane enclosed structures and a nucleus. And second, to describe the kinds of membrane enclosed organelles contained in eukaryotic cells. So by way of a quick review, let us remind ourselves about a couple of major core concepts that we have learned in units 1 and 2. And that is, first, cell membranes are composed mainly of lipids, proteins, and carbohydrates, organized in complex arrangements to perform a large variety of functions. We focused much of our attention to the plasma membrane in Unit 2, and there we saw that the plasma membrane acts as a selective barrier that controls the movement of molecules such as nutrients and waste products between the inside and the outside of the cell. These are the two core concepts that we learned thus far. And here in Unit 3, we will explore more elegant functions of cell membranes, first by looking at internal membrane systems, or otherwise called endomembrane systems, here in Chapter 5. And this will be followed by an examination of the key role of plasma membrane in cellular signaling in Chapter 9. Let's revisit a little bit of history from Unit 2. Our deep understanding of life has been driven primarily by our mind's insatiable curiosity. But this deep knowledge was, of course, realized practically through our ability to create new tools that extends our power of observation beyond what we can see with our naked eye. So the previously unknown living world in the realm of micron scale, or the so-called micro world, existing at the 10 to the minus 6 meter, or a millionth of a meter sizes, came about through the work of two men. In 1665, Robert Hooke built a new device called the microscope that he used to observe thin sections of dried cork from plants as shown in this figure. In these sections, he observed small cavities and named them cells. Later in the 17th century, Anton van Leeuwenhoek improved the magnifying power of microscopic lenses, and scientists were able to see unicellular organisms, including bacteria, protozoa, and algae. By the way, if you are interested in seeing a perfect replica of all of these really cool microscope that was originally invented by Robert Hooke and Anton van Leeuwenhoek, you can visit the Corning Museum of Glass, and there is a fine exhibit of these rare specimens there. These marvelous observations into the teeming microworld of life led to what we call as the cell theory, which was formulated in around early 1800s together by three German scientists, Matthias Schleiden, who is a botanist, and Theodor Schwann and Rudolf Virchow, which are both physicians. There are three fundamental near-universal tenets to their cell theory, I must say. One is all organisms are made up of cells. Two, the cell is the fundamental unit of life, and cells come only from pre-existing cells. So with these statements of the cell theory, I would like you to consider the following. First, if all organisms are made up of cells, and the cell is the fundamental unit of life. Are COVID-19 viruses, which are responsible for the pandemic that we are now confronting, are they considered living entities? Are they alive? Number two, if cells come from pre existing cells, the question is, how did the first cell originate? So these are pretty challenging questions that are whose answers are debatable, and this underscores, of course, the difficulty of defining what life is. Now, it is important by now to appreciate the fact that there is an additional tenet in cell theory, and this arose from modern biochemical observations that cells are defined by membranes. Careful and systematic microscopic observations of cells, coupled with biochemical analysis, revealed that there are two basic types of cells based on fundamental differences in organization: the prokaryotic versus eukaryotic cells. As depicted in this figure, all cells have a plasma membrane, but eukaryotic cells also contain membrane bound regions within the cell. A eukaryotic cell can be compared to a large factory with many rooms and different departments. And each department has a specific function and internal organization that are exquisitely coordinated with other departments. And collectively, they contribute to the overall quote unquote life of the factory. Therefore, what we call life is an emergent property of the cooperative and synergistic interactions of the various well-organized membrane-bound departments within the cell. This is a very important principle that we need to appreciate. The emergent property of the various parts, complex parts of the cells cooperating together, and generating a synergistic. When we use the word synergistic, it means a, uh, a property that you don't find in the individual components but come about only through interaction. So this is a fundamental property of life at all stages of organization. So this table here provides a very succinct summary of the difference between prokaryotes and versus eukaryotes, as depicted in the previous slides. So if you compare prokaryotic cells and eukaryotic cells side by side, you can establish a few criteria to differentiate between them. So first is with respect to the presence of nucleus. Prokaryotic cells do not exhibit a distinct nucleus or nuclei, but instead display what we call as nucleoid region. It's under the microscope. It's kind of a densely staining area that um, is an indicator of where the DNA is. Whereas in eukaryotes, you have, of course, a distinct nucleus. In terms of transcription, where it is located, transcription, as we know, is the term for the synthesis of RNA. In prokaryotic cells, this takes place in the cytoplasm, whereas in eukaryotic cells, they take place in the nucleus. The location of translation, or where the manufacture of um, proteins take place in prokaryotic cells, they take place in the cytoplasm, and in eukaryotes, they also take place in the cytoplasm. Um, with respect to the cell membrane components, they are also a little bit distinct. In terms of composition, the prokaryotes have what we call as hopanoids, whereas in eukaryotes they have sterols or cholesterol as part of their cell membrane. Of course, they all share the same phospholipid membranes components. In terms of size, the prokaryotic cells are really small, from 1 to 2 micrometers, for example, with E. coli. The eukaryotic cells, on average, is 1 magnitude larger, or 10 times larger, from 10 to 20 micrometers, or they can even extend up to a meter long, or even longer. So eukaryotic cells are obviously display wide range of sizes, much larger than prokaryotic cells. So with respect to the ratio of surface area to volume, the prokaryotic cells exhibit high ratio compared to eukaryotic cells. And with respect to internal organization, this is one of the most distinctive difference between prokaryotes and eukaryotes, is that prokaryotic cells exhibit no distinct organelles. Some contain circular structures, small molecules of DNA called plasmids, but they do not contain a membrane or distinct membrane-bound organelles, whereas eukaryotes contain various kinds of membrane-bound organelles. Let's take a closer look at eukaryotic cells. In this particular slide, we have an average or typical animal cell. The word typical is a misnomer because there is no such thing as a typical animal cell. It's really hard to find a typical animal cell in the face of huge diversity in its architectural patterns. So, but for the sake of simplicity, we can highlight major parts of a typical quote unquote animal cell. The first thing is the endoplasmic reticulum. This particular organelle is involved in the synthesis of proteins and lipids. And whatever is synthesized as proteins and lipids are then modified by the Golgi apparatus, which then act as a sorting station so that these molecules move to their final destinations. There are also special organelles called lysosomes, and lysosomes contain enzymes that break down macromolecules such as proteins, nucleic acids, lipids, and complex carbohydrates. And you have seen in Unit 2, of course, a very special organelle called the mitochondria, right there, which are specialized organelles that harness energy through cellular respiration in order to supply them with energy for the cell's needs and function. And I would also point out the remarkable structure called the cytoskeleton, which is a protein scaffold that is very dynamic and provides cell shape and structure. Of course, we have the plasma membrane right here, and the main largest organelle, which is the nucleus that contains the genetic material, the main genetic material. Later on, we will see that other organelles, such as mitochondria, and in plants, the chloroplast may also contain DNA. Okay. It's also important to note that the nucleus is the site of where RNA synthesis or transcription take place, as we have already mentioned earlier. Now let's look at a plant cell. In a plant cell, many of the organelles are the same as those in the animal cell. However, there are some outstanding features that are quite unique to the plant cell. And the first is, of course, the presence of a cell wall. These are present in all plant cells, and it provides additional support to the cell that are situated outside of the cell, the plasma membrane. And there are also the vacuoles. The vacuoles are huge In fact, they are the largest organelles in plants, and they vary in size depending on the maturity of the cells. So younger cells have smaller vacuoles, but older cells have larger vacuoles, and they contribute to the structure of the cell by maintaining turgor pressure by absorbing water. And so they confer stability to the cell by exerting, by the aqueous um, um, content exerting force against the cell walls. Chloroplast is, of course, a very unique organelle in plants that convert the energy from sunlight by the process called photosynthesis into chemical energy that the cell can use. There is also a very interesting structure in plant cells called plasmodesmata, which is a continuous connection between neighboring cells so that effectively the cytoplasm of each individual cells are interconnected with each other in their neighbors. So effectively making the cytoplasm of plant cells um, physically continuous to one another and facilitates in the movement of a molecule between one plant cell to another. The entire contents of the cell minus the nucleus make up what we call as the cytoplasm, and this is of course the same in animal cells. And there is also another term that you may want to remember called the cytosol, and this is the region outside the organelles but inside the plasma membrane. Okay, and again, this is also true for animal cells. So cytoplasm is the contents of the cell minus the nucleus, whereas the cytosol is the region outside the organelles but inside the plasma membrane. It's the soluble parts of the cells outside the organelles. So this concludes our overview of cell structure and function in prokaryotes and eukaryotes, which should provide us a solid framework into a more detailed exploration later on of cellular organization. So before you end, please answer the REEF question in the next section. And I hope that you will get the fundamental points with respect to the difference between the eukaryotic and prokaryotic cells before you answer the REEF questions. Okay, so thank you, and I'll see you in the next lecture video, video 5.4.